Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about how the genetic code was cracked. And when I say the genetic code, I mean the sequence of nitrogen bases and how it was figured out in terms of what the nitrogen bases meant in terms of a polypeptide sequence of a protein. Now, on on surface, when you talk about that, it's like, well, that is that significant? Well, of course it's significant. It's cracking the genetic code. We're talking about one of the great discoveries of all time in biology. And so, what I want to do is uh, get into this conversation in terms of a story. And I want to describe some of the experiments that led to cracking the genetic code. Because I'm going under the assumption that we are familiar with the fact that the genetic code is located on messenger RNA, and that's translated by ribosomes in the cytoplasm of both prokaryotes and eukaryotes. And going under the assumption you know that. And I'm going under the assumption that you know that it's a three-letter codon that corresponds to each of the 20 amino acids. But I want to talk to you sort of like, again, a story of some of the scientists involved in cracking the code. Because the reason I want to do this, and I want to introduce you to the Nobel Prize winning scientist Marshall Nirenberg, who is instrumental at cracking the genetic code, is because I think at this stage in my career in biology, I find it more valuable to get into sort of the head of the scientists and sort of the methodology and research design more than it is the content of the material. I'll apologize for that. I'll update that later. And so it's more important that you understand that the information found in textbooks or on the internet is really the result and the conclusions of some of these great experiments. And so this is I'm not sure if there's many great experiments, but this is certainly one of them, and I hope I can give it justice. And so Marshall Nirenberg had this dilemma. In other words, when you look at RNA, and this is a single-stranded molecule of RNA polymer, and you notice the nitrogen base is sticking out, the question is, how is the genetic code interpreted? So it's, in other words, like the nitrogen base sequence is the genetic code, but like what is the code? In other words, I mean, we like to think that maybe it's a single, like cytosine stands for one of the amino acids, and guanine is another amino acid, and uracil is another amino acid. And what did I leave out? Cytosine, no, guanine, and adenine. Adenine was one of the one of the amino acids. But as it turns out, these are just four, when in fact a living organism has 20 different amino acids. So it can't be read one base for one amino acid. And so here's a picture of all 20 of the amino acids. Here's the amino group over here. Here's the carboxylic acid. So all of them have this fundamental same structure. What's different about them is the R group. So glycine, alanine, valine, leucine, isoleucine, etc. So there's 20 different amino acids. And so I know what you're thinking. You're like, well, perhaps if it's not read every base for one amino acid, maybe the genetic code is read in groups of two. And so you go 1, 2, 1, 2, 1, 2, 1, 2. But if you read the four nitrogen bases in groups of two, you'll get only 16 possibilities, and that's not enough either. And so let me sort of just show you, like AA or AU or AC or AG. And then if you continue to this, GG, GU, GC, and GA. If you keep doing that, there would only be 16. So the genetic code is read in groups of three. 1, 2, 3, and 1, 2, 3, and 1, 2, 3, and 1, 2, 3. So it's every three is the codon that corresponds to an amino acid. So if you read the genetic code in groups of three, that gives you 64. So that's 64 possibilities. So that's even more than you need. And so we'll get into that a little bit later in the conversation. So the genetic code is read in groups of three, but here's the question. What is the genetic code, and how is it translated into, in other words, what's the genetic code for glycine? Is it CAG? Is it UAG? In other words, what's the genetic code for phenylalanine? What's the genetic code for lysine? So how could you design an experiment to figure out the genetic code? In other words, crack the genetic code. It's kind of interesting, huh? So why is this relevant? Well, each protein has a unique sequence of amino acids, and that's known as the primary structure. So the sequence of amino acids. And so the genetic code in messenger RNA and then obviously in DNA holds the recipe for the sequence of amino acids. And why is that important? Because the primary structure of proteins ultimately determines its three-dimensional folding, and its three-dimensional shape determines its functionality. So this is hugely important. Okay, so how are we going to do it? Well, I'll talk to you again about this. And so we now know, how the genetic code is read. For example, AUG happens to be a start codon. It's the start of most proteins. And it also is the codon for the amino acid methionine. And ACU is for threonine. And GGC is for glycine. Okay, so you're like, well, I'm revealing the code. Yes, I'm revealing the code. I'm just showing you that we do know it. But I want to know how we determined it. That's what I want to talk about. And so I hope this is intriguing you. So Marshall Nirenberg, upon discovering the genetic code, gave a seminar describing the assay in which he went about doing this. And so in 1961, as it turns out, kind of curious that he gave this presentation in Moscow, and it wasn't well attended considering the magnitude of the discovery. Okay, one of the great discoveries of all time, I'm telling you, in biology. But perhaps it was the name. Remember this: if you're ever giving a presentation or writing a book, be careful about calling it something standoffish or intimidating. So he called the title of his presentation "The Dependence of Cell-Free Protein Synthesis Upon Natural and Cell-Free RNA." And apparently that wasn't just bringing in the huge crowd. But as it turns out, as we'll see, when you understand the experiment, this is actually a pretty accurate title. And so this was the seminar that cracked the genetic code. So let's get into that. And so what he went about doing was, this is a description of the experiment or the assay. So Marshall Nirenberg took some E. coli, some bacteria, okay? And so hopefully you're familiar with that. These are prokaryotes. They have one chromosome of DNA, and of course transcription produces RNA, and they have ribosomes. And that's pretty much it for a prokaryote. You get these ribosomes, and of course ribosomes are where messenger RNA is translated into protein. So this is what Marshall does. He takes the bacteria and he puts them in a blender, and he's like bzz, 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 and he breaks them down. In other words, breaks down the cell wall and cell membrane, makes something like, so this is the extract, we're going to call it, sort of like a bacterial soup. And so you're like, hold on, what is that? Well, again, these are bacteria, so this is a prokaryotic cell. If you put it in a blender, you're going to break the cell walls down. You can use other techniques. You can use enzymes called lysozymes and freeze and break open bacteria. It's commonly done. But you could break the bacteria open, and then you could also add an enzyme that will degrade the DNA. And as it turns out, he went ahead and did that. And so basically what the extract is cytoplasm, and you've got ribosomes. And so you got ribosomes in here. And so ribosomes, again, just to clarify this, to make sure you're following me, a ribosome, which is this guy right here, reads messenger RNA, reads RNA, and it's the one that contains the genetic code. So in other words, the ribosome is reading the genetic code in groups of 3 and attaching the amino acids together, forming peptide bonds to produce a polypeptide. So proteins are being made on ribosomes. And so, okay. So what does he do? He takes the bacteria, he breaks it down, and so you got ribosomes in here. Okay. So these are the ribosomes. And then he adds to this E. coli extract, he adds some radioactive amino acids. So he's able to do that, kind of clever. Okay, so let me just get into that. So these amino acids are radioactive, so they can be traced. Okay, right there. So radioactive amino acids, he's adding these. And so whatever this juice was, and we know what it is, it's a bunch of ribosomes. Whatever the juice is, the extract, it can make proteins. So in other words, it can take these radioactive amino acids and link them together and make a protein. And you're like, huh, okay, that's interesting. So this is what he does. He blenders the E. coli. He makes an E. coli extract. He adds amino acids to it, and they make protein. Okay. So what's really cool about this, though, is if you take this extract and you put it in an incubator, 37 degrees, and you let it sit in there for about a half an hour, about 30 minutes, okay? And then you do the same thing. You add these radioactive amino acids. So there you are adding those radioactive amino acids, and then lo and behold, there's no protein. So a negative result, that's kind of weird. And so you might be thinking, I don't know, maybe it's the temperature, but it turns out that it had nothing to do with the temperature. And what's interesting about this is that if you put it in the incubator just for 5 minutes, say, it is capable of producing a protein. But if you wait approximately 30 minutes and add these amino acids, there's no protein. So that's kind of strange. Something to think about. Okay? So it's a negative result when you let it sit around for 30 minutes. 45 minutes, no protein. An hour, no protein. 2 hours, no protein. But about 5 minutes, it's still capable of making a protein. Okay? So as it turns out, what's even more cool about this, a little more interesting, is that you take the same E. coli extract. Okay, so same thing. You blender the E. coli, so the ribosomes are in here. And then you're adding your radioactive amino acids to it. Okay? You let it sit for 30 minutes at 37 degrees. And you're like, well, it's not going to make a protein. Ready for this? If you then add RNA, okay, if you add RNA from a different E. coli, let me do this properly, a different E. coli. So in other words, exogenous RNA, so free RNA. If you're adding free RNA to this, guess what? It'll make a protein. So what do you make of that? It's like you thought that the extract wasn't capable of doing it after 30 minutes, but then you add RNA and it is capable of making a protein after all. What are your thoughts on that? Okay, you can pause the video and ponder this for a little bit if you felt like it. So this is a positive result. This is a little weird. But the implication of this is huge. Okay? The implication is, what's happening during the wait? What's happening between 5 minutes it can make a protein, 30 minutes it can't make a protein, but then when you add RNA, free piece of RNA from a different group of E. coli, you can make a protein. The only logical conclusion is that somehow the RNA from the E. coli that you were working with in the extract degrades. In other words, it's no longer present. And we now know that about RNA, is that it's short-lived. There's RNases that break down RNA very rapidly in the cell. And that's to the advantage of cells. You don't want messages around in the cytoplasm that are old. So you want to, today's news is important. Make that protein, but then get rid of that message. And so it's a way in which the cell can be efficient in terms of protein synthesis. So RNA doesn't last very long in the cytoplasm. So in the E. coli extract, RNA is broken down. So therefore, you can add all of the amino acids you want to your merry content, and there would be no protein. You need RNA. So if you add RNA to this, the ribosomes are fine. The ribosomes are fine. And so you're like, okay, what's the big deal? How's this even cracking the genetic code? This is huge, because you know what we've just been able to do? If you can take E. coli, so what we're saying here is, if you can take E. coli, make an extract, okay, let it sit for about 30 minutes, then it's basically ribosomes capable of making a protein if you give it RNA. So you have something that paramount to the fact that the E. coli extract can be, if you will, programmed to produce a protein if you give it a piece of RNA. So in other words, it's like a DVD player. It'll play whatever DVD you put in, or CD player, it'll play whatever you put in. Any RNA that you put in, it'll produce a protein. And so this is huge, because, so here's the thought, that if you give the E. coli extract a simple enough RNA, now the truth be known is, back here, when you added this exogenous RNA, if you add a big piece, it'll start making all kinds of proteins, and there's no way to keep track of it. But if you add a very simple and small enough piece of RNA, you might get a sense of what the amino acids are for that piece of RNA, and you might be able to crack the genetic code. And so Marshall Nirenberg is quite excited about this. He's so excited is that he flies from his lab at the NIH in Maryland, he flies to California, and he meets up with a very impressive scientist studying at the University of California, Berkeley. Wendell Stanley, you might be familiar with Wendell Stanley, who is working with tobacco mosaic virus. And tobacco mosaic virus is a simple rod-shaped virus with a protein coat and a single strand of RNA. And so you can imagine what Marshall Nirenberg is interested in. He's interested in working with Dr. Stanley, because Dr. Stanley has this fairly simple piece of RNA, and maybe they'll use this to crack the genetic code. But they worked at this quite feverishly, but all the experiments failed, and won't get into that, but it's kind of complicated in terms of the way in which the virus actually replicates its RNA, and so the experiment was kind of doomed to fail. It wasn't going to work out. So meanwhile, when Marshall Nirenberg is in California trying to battle with the tobacco mosaic virus, check this out. Back in Maryland, one of Marshall Nirenberg's graduate students, Heinrich Mattei, okay, he was back at the NIH in Maryland, and so most of the E. coli and the radioactive amino acids and the RNA, Marshall Nirenberg took to California to continue in the experiment. But Mattei had just a few radioactive amino acids. He didn't even have all 20. He just had a few of them. And he didn't even have a large bank of RNA to work with. He just had some simple pieces of RNA, but that's all you really needed. Some simple pieces of RNA that apparently they were just using as controls in their experiments when they were working together in the lab. So this is a picture of Marshall Nirenberg and Heinrich Mattei. And so as it turns out, late one evening, as the story goes, Mattei made his E. coli extract, and what he did was he added a very small piece of RNA to the E. coli extract, and it was a polyuracil piece of RNA. In other words, it was continuous U like this, so it's poly-U. And some of the amino acids, I think it was about 6 different amino acids. Now, at this point, you get lucky, because you don't know what this is going to code for. It may not be one of those 6, but he got lucky. He didn't even have all 20. So as it turns out, is when he added this to the extract, this poly-U RNA, like this. So he added RNA, like this. And he added some radioactive amino acids to this. After letting it sit for 30 minutes at 37 degrees, he started to get a small little polypeptide to form, and it was radioactive. And so he got a positive result. And it's like, what amino acid is this? And what amino acid is this? And what is this? And what is this? Well, if you're following this conversation, and understanding it, you'll know that the genetic code is 123 and 123 and 123. So in other words, what is that amino acid that is the genetic code for you? And guess what? The genetic code for you, when translated, is the amino acid phenylalanine. And so what did Matai get? He got this little tiny poly, not very big. I would question poly, I would say more oligo. But oligophenylalanine. He got this little tiny phenylalanine. And guess what? That must mean that the genetic code for the amino acid phenylalanine is U. It's huge. So that was the very first codon deciphered. And so, how do you like this? What do you think you would do next? Okay, so then maybe instead of adding U, you would add like a poly A RNA and see what that codes for. And then maybe a poly G, guanine, or polycytosine. And so, so Marshall Nirenberg returns to Maryland, and they continue experimenting. And it's more difficult to determine the other codons. I'm not going to get into that conversation in this video, but there's more elaborate techniques that are needed to require the decoding of more complicated amino acid codons, such as like AUA or CGA. But nevertheless, in the early 1960s, in mid 1960s, the genetic code was deciphered. And so this is a picture that we can use today to figure it out. So in other words, let me just walk you through. So if the very first codon was U, right, and that was for phenylalanine, so this is considered to be first base, second base, and third base, like that. So who's on first? So who's on first is uracil, and then second base is uracil, and third base is uracil. Guess what? It's phenylalanine. That's right. So that's the amino acid phenylalanine. Let's just go, how about CUG? What's that? So C, U, and then G. CUG is leucine. So this is the genetic code. Check it out. It's pretty cool. Remember we said that it was 64 codons when you have 4 nitrogenous bases and they're read in groups of 3? Well, as it turns out, the code is really for 61 amino acids, because there's going to be 1, 2, and where's the other one? Oh, right here. So there's 3 stop codons, UAA, UAG, and UGA, or stop codons. So only 61 of the codons code for amino acids. And as it turns out, AUG, let's just look at that, AUG codes for the amino acid methionine. Methionine is kind of cool. It has sulfur as in its R group, and methionine as well as cysteine are the only ones that have sulfur in them. And so it also codes for the start of translation. AUG is the start codon. So here's another diagram of the genetic code. You can find these on the internet if you're interested in it. Now, the genetic code has to be read in a particular orientation, as one can imagine. You can't just read things forward and backward. So they're read in a 5' to 3' orientation. So it would be AUG, and I'll just make up one, CCC. So AUG, so it'd be A, first base, U, G is, hey, methionine. That would be the first amino acid. And then CCC would be, who's on first? What's on second? I don't know. Who's on third? Is proline. Okay? So there it is. That's how you use the decoder of life. And so you can find all sorts of different ones. And I wanted to point out the genetic code has redundancy. Look at this. Look at all the redundancy for the amino acid serine, or proline, or alanine. But it's not ambiguous. It's not random, but it is indeed redundant. Okay? So any one particular codon does code for one amino acid, but you can't go in the reverse direction. So in other words, you can't say, I know that the amino acid is proline, so therefore the genetic code is, you wouldn't be able to determine that. So you can't go in the reverse direction. So the as it turns out, that what's significant is the very first two bases can almost do it. It's the third base that is kind of um, wobbly in terms of it can fluctuate and still determine the same amino acid. And that's important too, because if there is a mutation, a change or a point mutation, a change in a nitrogenous base of DNA, if it happened in the third base of a codon, it's less significant. It could actually occur and there'd be no change because it would still code for the same amino acid, so it would be considered to be a silent mutation. So that's pretty significant. And so just to make things clear is that to establish the reading frame, the codons are read in groups of 3, as we discussed. They're read in a 5' to 3' orientation, and they're non-overlapping. Okay, so that's important. And so the really, really cool thing about the genetic code is that it's universal. And if it's universal, that means that all organisms share this genetic code. And so you're like, well, how do we know this? Well, we know this, again, through experimentation. This is a picture of a tobacco plant. And I don't know if you are familiar with tobacco plants, but I can tell you they don't normally glow. If you talk about glowing things or bioluminescence, you might think a firefly is capable of glowing. It produces luciferase, which is glowing. As it turns out, you can take the gene or the RNA from a firefly and add it to a plant, and it'll transcribe that into a firefly glowing protein, and so therefore you can make tobacco plants glow. And so think about this. Think about the implication of this. Now, since the genetic code is universal, we could transfer genetic material, RNA and DNA, from one organism to another. In other words, genetically engineer, genetically modify. And so what we're talking about is the birth of biotechnology, and the implication of that is huge. And so in labs, genes can be transcribed and translated from completely different species. So here, again, is a tobacco plant expressing a firefly gene. And so we've taken these techniques, and here's a particularly important protein, insulin, life-saving protein hormone required by the body to reduce blood sugar. It's produced by the islets of Langerhans in the pancreas by the beta cells. And so some individuals are not able to make insulin, so we can give them insulin, but how do we produce enough insulin? Well, how about if we take the gene that codes for this polypeptide and put it into a rapidly reproducing bacteria, for example. And so we've done this, and it sort of was one of the key experiments in the birth of biotechnology. And so we can get organisms to read each other's genetic code to produce proteins upon desire, and then we would want to isolate that human protein from all the other bacterial proteins. So pretty cool. Now, when I say that the genetic code is totally universal and every living thing uses the same genetic code, not completely accurate. There are a few exceptions, like this paramecium, oddly, does not use the same genetic code. Not sure why. And of course, there are some exceptions in the genetic code of DNA in mitochondrial DNA and in chloroplast DNA. There's some different codons involved in those two organelles. That's kind of curious. But nevertheless, the universality of the genetic code clearly has been operating for many years, since all living things have it. It's been happening. And so this sort of shared genetic vocabulary, if you will, is a reminder to us all of the kinship and bonds between all living things on the planet Earth. And here's a picture of one of the great biologists of all time, Jane Goodall, and some of her amazing observations of chimpanzees, showing the world that how closely we are to chimpanzees, and how all of life on the earth needs to be respected. I wish, not to get too sappy about this, but I wish sometimes that Charles Darwin would be able to come back and understand some of these great experiments that we've been talking about in terms of the genetic code and the common link between all organisms. he had a notion about all organisms descending from a common ancestor, and I can't think of any more conclusive evidence to support that than a common genetic code between all living things. So there you have it, cracking the genetic code. I hope you enjoyed it and learned a little something about our genetic code. Thanks for watching.